So thank you very much, dear audience and organizers of this interesting workshop. So this is to provide you in a nutshell an overview of the Marlon project. You may have noticed that just as the Gijs project, Marlon is also a very familiar first name. Not just of famous actors like Grace Kelly and Marlon Brando, but also, I guess, commoners. But okay, so the schedule is actually to provide some background and to explain about the project and to also feature some conclusions briefly. So before I start off, I'd like to present this this video. Actually, it was a presentation by the head of the French Haut Conseil de Biotechnologie, which is a kind of risk assessment council, and there was at that moment a discussion within the French Parliament, following the publication of the famous study by researchers from Caen, Ciarlini et al., which reportedly showed tumor formation in rats fed NK603 mice. And he very well captured actually, I think, the essence of both the Gijs and the Marlon project. And after this I can go home because he's actually told you everything you need to know about the project, I guess.
Si vous voulez tester un aliment complet dans lequel il y a des milliers de molécules, c'est beaucoup plus difficile. Ce n'est pas la même technologie. C'est tellement difficile que l'EFSA, actuellement, dans le 7e PCRD, que l'Europe, actuellement, dans le 7e PCRD, finance 2 projets pour répondre à ce type de questions. Le premier projet, c'est le projet GRACE, qui a démarré cette année et qui vise justement, dans le cas de toxiques classiques, à remplacer ou à trouver des solutions alternatives à des mesures faites sur des rats, sur des souris ou sur des cobayes, et qui voudraient utiliser des technologies in vitro, par exemple. Et l'autre projet est un projet Marlon, qui va démarrer début 2013, dans lequel on se pose la question de savoir comment suivre l'épidémiologie à long terme, c'est-à-dire une fois qu'un maïs ou qu'un soja ou qu'une plante transgénique est mise sur le marché, puisqu'elle est consommée essentiellement par des animaux d'élevage, déterminer effectivement sur le suivi de ces animaux l'apparition ou non de signes avant-coureurs d'une toxicité probable.
Okay, so thank you, Mr. Lagay, thank you very much. So next slide. So I realize I have to keep this very short. So those of you who are familiar with risk assessment, post-market surveillance and post-market monitoring is a very common term. I think this is the big difference with what we previously discussed. So the rat feeding experiments are usually performed before a product is brought to the market, at least in some regulatory constituencies like Europe, where there is a mandatory requirement. I'd also like to stress that, outside Europe, this is not considered a mandatory requirement. But okay, this is about pre-market assessment. We are talking about post-market monitoring. So usually a product will have been approved and then regulators will impose requirements for post-market monitoring. Why is that? Well, for each GMO that is allowed onto the European market, there's this requirement for general surveillance, and that is for unforeseen effects. But there's also the possibility in regulation foresees this possibility that actually coupled to the decision to allow a product onto the market, there may be a requirement for case-specific monitoring. And that means that for this particular product, a potential health effect may have been identified. And in order to verify whether this really occurs in the marketplace, monitoring should be performed. This is something that also actually can be combined with the next one, which is verification of assumptions. So in a number of cases, for instance, we've seen that soybean oils with a modified fat composition or oil composition were actually allowed onto the European market, but then Commission stated, because the modified oil composition can have an impact on the, say, nutritional status of consumers, Because, for instance, you have essential fatty acids like the polyunsaturated fatty acids, you have the monounsaturated fatty acids. They wanted to make sure that the estimates of intake of these oils made during the pre-market assessment, so when you have a kind of exposure assessment, but of course this is, based on certain assumptions because the product is not yet on the market. So you have these so-called substitution scenarios. So it's important to verify if the intake of the product is indeed what was expected. So this is something that is optional. It has been asked for food, like the soybean oils that I told you about, but not yet for animal feed, not for specific hazards identified or specific products for animal feed. However, policymakers want to be prepared, and for that purpose, it would be useful to have a tool available in case, applicants, which are the companies wanting to sell a biotech product, want to bring it to the market. So just in case, the Commission would say, well, if the Commission said, well, you can market this product, but we would like you to monitor for this or this particular effect in livestock. That also shows that we have a dilemma. We don't know on beforehand for which product, for which type of health effects, and in which animal species such a question could be posed, such a request could be made. So with the Marlon project, we actually tried to develop a methodology that would actually fit this knowledge gap, or at least this lack of a tool that the Commission could provide, standardized or harmonized methodology that the Commission could provide to applicants. So the Marlon project that tried to address this problem was funded by the 7th Framework Program. It started in 2012. It was a 3-year project. And it was a so-called CSA project, which means that no original research, like experiments in livestock, were carried out. So we did not go out into the field and started monitoring livestock, for effects of such things, or performed feeding experiments with animals. So we had to draw upon the information that was available and develop a methodology. So there's a lot of work, desk work, database development, epidemiological tool development, which is also a kind of statistical tool. Okay, so I will rapidly go through this, so I will skip a few of these slides here. So besides, of course, this purpose of monitoring, that it might have broader applicability. It might give you insights into relation between feed and health in more general terms. This is the logo of Marlon. We had 8 partners working together, or 8 countries, 11 partners in 8 countries working together on the project. Some of them are actually here in the room, so I'm quite happy. So if you have difficult questions, of course, I can delegate them to our colleagues. Like, for instance, our Bulgarian partner and our German partner. Actually, the activities within the project focused on 3 main topics. One was data collection, so what knowledge is already there, what has already been investigated on, the impacts of GM feed on animal health. So what initiatives are there that we could actually obtain data from, what monitoring initiatives. The model development, it's a kind of statistical tool that will actually help you to predict, whether, when you have a hypothetical effect of GM feed on an animal, you have an idea, like it could be an effect on, for instance, nutritional efficacy, or maybe some other kinds of effects. And you could say, for instance, well, this will impact on, say, performance by 20%. So then the issue is, if you have this effect of a certain size, could you pick it up with the current surveillance and monitoring systems against the background variability that we know that exists for this particular, parameter like performance. So actually the tool that we developed will help you actually predict whether with the current surveillance and monitoring programs you can set up a system to detect this effect, whether it makes sense to do so. And actually we drew upon, say, a rather fashionable, I guess, trend in the sense that we actually wanted to exploit what we call syndromic surveillance. So this actually refers to commonly measured health syndromes in livestock. Like, for instance, mortality in animals, morbidity, performance, carcass characteristics in the abattoir. So things that are commonly inspected by, for instance, a veterinarian visiting the farm, or perhaps an inspector of carcasses in the abattoir, or perhaps pathologists working in laboratories, perhaps people working in a dairy laboratory analyzing the milk that comes from the cows and such. So you need to have an hypothesis of an effect of the GMO and translate that to a commonly measured syndrome. We also performed what we call a feasibility study for that model, testing it in various scenarios, collecting data on various scenarios. We also had colleagues, for instance, our colleagues from Berlin, who actually investigated the characteristics of production chains. So what kind of data are collected inside production chains regarding animal health, for instance, regarding feed production, and what can be reliably measured, and what can be useful information for the purpose. And of course, the issue is that we have a wide range of animal species, production systems, and a question frequently raised is, whether you could compare organic systems with conventional systems, because organic is known to be non-GM it's a kind of principle there. But unfortunately, there are too many other confounding factors. Okay, so what I'd just like to highlight here in the next minute is a few highlights, the outcomes. We have developed an epidemiological tool, which is kind of statistical tool, that will help you predict whether or not you'll be able to identify health impacts. My colleagues from Bulgaria, they established a database where they actually used scientific literature, but it's special in the sense that, this literature in which GM feed was tested in animals, they singled out all the single parameters that were measured. So if you're looking for a particular effect or a particular health parameter, you can already get an idea of what has previously been tested. That will help you, of course, set up a monitoring program that need to look into those effects in future. And besides that, we also, published several deliverable reports, which we intend actually to publish as scientific articles in a special issue of a scientific journal. And we also hosted a training event, stakeholder workshop, and published recommendations and guidelines in an overarching report and a flyer. Okay, so these are just examples. So I hope I have still 1 minute left. Okay, so this is an example of how actually the statistical model works. It works what we call a risk scenario tree. So it considers that there is a kind of chain of events. So we have a farm here. This is a dairy farm. So you have dairy cattle here. It's very abstract, of course. Now you may have a sick animal that turns sick. For instance, by consuming a certain feed. So in those cases, you might have clinical signs that are present or that are not present. If clinical signs are present, the farmer may detect them or may not detect them. If the farmer detects them, he calls out a local vet. Well, the veterinarian will inspect the animal, will collect samples, and will have them analyzed. So in each of these steps, there may be a detect or a non-detect. And if you start to simulate this, with data collected, for instance, from databases or from experts, soliciting expert feedback on, the likelihood of all these occurrences, then you can actually perform the kind of Bayesian probabilistic simulation, as I call it. So trying to simulate with a statistical tool how likely it is at the end that if an effect occurs here with a certain intensity, that in the end you will see a change in the trend of reported effects, Okay. Okay, so we do have a website. The database does have a website. So I'll be very brief about the conclusions. I think that one of the main outcomes and the reason why we actually developed this tool was that technically spoken, we tried to look at it from different angles. There was no real possibility to measure prior exposure to GM feeds. You know, when you have an animal on a farm, it's very difficult to tell what quantities of GM feed he has been exposed to in previous years. There were suggestions actually for an alternative approach, alternative to this, to have what we call case control studies. So you pick, for instance, two farms that are very similar, and then you have GM crops being fed to the animals on one farm and not to the animals on another farm, and then you can compare both. But those are controlled studies, and the model developed here is actually for what we call non-controlled conditions, so where you actually go into the field and collect data. Okay, so I hope I haven't exceeded the time too much, but okay. Thank you very much.